Hello everyone, it's Tutor Mifzal here. Today we will be discussing about biology, SPM, carbohydrates. But before we do that, do follow me on my Facebook and Instagram account at Tutor Mifzal. And don't forget to like, share and subscribe to my YouTube channel. So now, let's begin. There are three types of carbohydrates: monosaccharides, disaccharides and polysaccharides. And the basic units of carbohydrates are monosaccharides. So they are monomer which can be linked by condensation reactions to form disaccharides and also polysaccharides. The most common monosaccharides is the 6-carbon sugar or hexose with the formula of C6H12O6, or as we all know, it is glucose over here. So the examples of monosaccharides are glucose, which is found in plants and fruits; fructose, which is found in sweet fruits and honey; last but not least, galactose, which is found in milk. So all monosaccharides are soluble in water, taste sweet and also can form crystal. And all monosaccharides are reducing sugar. As I have mentioned before, glucose, fructose, galactose, meaning all monosaccharides can combine together to form disaccharides and also polysaccharides. Disaccharides meaning that it consists of two monosaccharides that combine together by a condensation reaction. So a condensation is a process where two small molecules are combined together to form a larger molecules by removal of one molecule of water. As such, we know that disaccharides are soluble in water, taste sweet and form crystals. In the case of disaccharides, most of the sugars in disaccharides are reducing sugar except for sucrose. Whereas in monosaccharides, all of the sugars are reducing sugars. So the examples of disaccharides are maltose, lactose and sucrose. So maltose over here is formed from the condensation of two glucose molecules. One glucose plus another glucose would get maltose plus water, whereas lactose is formed from the condensation of glucose and a galactose molecules, whereas the condensation of a glucose and a fructose molecules would form sucrose. However, we can change maltose back to glucose by a process known as hydrolysis. So hydrolysis is a breaking down of a large molecule to two simple molecules by addition of one molecule of water. Hence, not only maltose can be converted back to glucose, but lactose and sucrose can be converted back to glucose and galactose and glucose and fructose respectively. So maltose is found mainly in germinating seeds, whereas lactose is mainly found in milk. Last but not least, sucrose is mainly found in sugarcane and sugar beet. All of them are reducing sugar except for sucrose. Sucrose are non-reducing sugar. Next, we have polysaccharides. So polysaccharides are polymers, meaning that they are formed by condensation of hundreds or thousands of glucose monomers. And the general formula is C6H10O5. And polysaccharides can also be hydrolyzed to monosaccharides by heating with acid or by enzymatic reactions. And polysaccharides are insoluble in water, do not taste sweet and also cannot be crystallized. So there are three main types of polysaccharides. The first is starch, which is found in wheat, rice, potatoes, bread and also corn. The second one is glycogen. The glycogen is found in animals, stored mainly in the liver and muscle cells and also fungi. In the human body, we also store our food or our excess food in glycogen form. Last but not least, we have cellulose. So cellulose is the material that forms the cell wall of a plant cell, and it is mainly tough, rigid, strong and flexible, and it also prevents the cell wall from bursting. So in order for us to test starch, what we would do is that we would test starch with iodine solution. Just a recap, the brown color of iodine solution turns blue-black when starch is present. So now let's look back at reducing sugar. We know that all monosaccharides and disaccharides are reducing sugar except for sucrose. And reducing sugars are sugars that can act as a reducing agent. To further understand the concept of reducing sugar, first we have to look at the experiment to determine reducing sugar, meaning the test that qualifies if a sugar is a reducing sugar or not. So what will happen is that when a solution of sugar is heated with the Benedict solution, the formation of a brick red precipitate indicates the presence of reducing sugar, meaning that if there is no brick red precipitate, therefore reducing sugar is not present. So a Benedict solution would turn brick red precipitate if reducing sugar is added. The reason being is that Benedict solution contains copper 2 sulfate, which is blue in color. So copper 2 sulfate contains Cu2 plus ions, which is blue in color. So what will happen is that the reducing sugar would reduce the Cu2 plus into Cu plus. So guess what? The color of Cu plus is the color of brick red. Therefore, brick red precipitate is produced. So now you might be asking me, what about sucrose? Do we have any indicator to show that sucrose is a non-reducing sugar? It's the same thing. If we add sucrose solution into a Benedict solution, then nothing would happen. So that is how we differentiate between reducing sugar and also non-reducing sugars. So that's about it for today, guys. If you find this video really helpful, do follow me on my Facebook and Instagram account at Tutor Mifzal, and don't forget to like, share and subscribe to my YouTube channel. See you soon in another video, guys. Bye!